I'm Endika Blanco. I'm doing my PhD in ICI4, Madrid, about the molecular response to grafting, analyzing the genotypes of Pinus pinaster with contrasting draft response. First, I have to say that this PhD is part of a larger project that aims to learn more about the mechanism behind the response of conifers. In this case, Pinus pinaster, to which the effect of water stress and the communication of this response between organs, for which grafting is a key methodology that we use. However, my PhD is focused on improving the understanding of the response of Pinus pinaster to the stimuli of grafting. First, I'll explain briefly what grafting is. Graft is composed in two parts: the needle, a tissue originated from another individual or another part of the same individual, that is attached to an already settled individual, the rootstock. Well, the grafting traditional use have been primarily used as vegetative propagation method in agriculture in order to enhance the production of fruit or wood, for example, taking advantage of the features of the rootstock, like more resistance to climate or pathogens. Nowadays, however, grafting is also used as a methodology to study the communication between different organs. Well, in order to study the water stress, the project works with well-genotyped and phenotyped individuals of Pinus pinaster, which are GAL 1056, native from the northwest of the peninsula, lives in a wet climate and has a great growth, and Oria 6, native from the southeast of the peninsula, lives in a wet climate and a dry climate and has a limited growth. And then we have the progeny of these two individuals, and from them we taken the most tolerant and the most sensible to water stress in order to do further studies. As I said before, in order to understand the communication of the response to water stress between the organs, the grafting methodology is used, and my PhD is focused on understanding and separating the effect of grafting from the effect of water stress. In order to achieve that, we use three grafting methods: not grafted, which we use the progeny not grafted, the progeny self-grafted, and the progeny rootstock grafted with GAL 1056. Then we applied that methodology to both the most tolerant and the most sensible of the progeny, and we taken a sample from needle, science stem, rootstock, and root. Then we extracted the messenger RNA from the samples and built a de novo assembly transcriptome. We use BLAST, InterPro-Scan, and EGNOM Mapper for annotation and functional characterization, and worked with DSEQ to analyze the transcriptome data in order to identify which genes are involved in the response to grafting. Within the preliminary results, we have the PCA just to check which variables have more strength in the variance of the data, which are primarily the genotype and secondly the variant, the organ. Then we made a differential expression analysis from which we extracted the differentially expressed genes for the grafting, comparing primarily not grafted versus autografted. And also we made through a weighted co-expression network analysis, we identified which genes modulus are correlated with each of the grafting types that we studied. And that's what's done until now, but there's a lot more to do. A complete functional characterization of genes due to the lack of characterization of the genes in conifers. an analysis of small RNA profiles, and analysis of hormone profiles. Thank you.